Good morning.
Okay, all right. There's going to be a bit of a lag on the response. Okay. What I thought we'd do is I'll tell you a little bit about what I am or what I do, how I got here. We'll just do a quick introduction. And then I've got a couple of questions for you about maybe what you think it is that I do. And then we'll see if we can actually get sort of a conversation going back and forth. So my name is Dr. Thomas Merritt. I'm a professor at Laurentian University. I have been here 8 years as of last week. So I got here in July of 2008. And I am a tenured full professor. So I was talking to your teacher a little bit yesterday, and he said one of the questions that you had was trying to understand what professors do. And what I eventually would like to talk to you about is what I do as a researcher and what genetics is and what genetics means to me, what genetics might mean to you. But maybe we could start by just saying what is a professor. So just give me a show of hands. How many of you think you understand what a professor does? Okay, good. So we're starting with an honest response. That's a good start. Okay, so how many of you think that as a professor, I do research? Okay, so we got a bunch of hands. That's good. And I do. Okay. How many of you think that I teach students? We get some hands. Okay. So I do. How many of you think that I spend a ridiculous amount of time in committee meetings trying to run the governance of a university? Okay, so that's a third of my job. And that's one that I think students, the teacher knows. I think that it's one that students have a harder time understanding. But when you get hired as a professor, you're evaluated every year based on three things. And those three things are your teaching, your research, and your governance to the university. And I've done a pretty good job of selling to my university that what you and I are doing right now counts as governance. So when I interact with students and tell you about what it is to go to university, the university sort of counts that as some of my governance work. But the teaching itself, sorry, the teaching itself is sort of interesting. I teach classes, and the classes are sort of similar to what you do in your own class, but I also teach graduate students. So when I ask you if I do research, I very rarely actually get to go in the lab anymore, get my hands dirty, and do research, but my students do. So I have graduate students that have finished an undergrad degree and they're coming back, and they actually physically do the work in the lab, and that counts also as teaching. So I'm mentoring those students and I'm working with those students. So the professor's job, you end up sort of split between working with students, research, those two are interwoven, and then a certain amount of committee work that almost nobody really wants to do. But if you're really fortunate, you can convince your university to count the kind of teaching that you and I are doing right now as governance, and that's a lot more fun. Okay, so that's sort of what my job is, and we're going to meet a couple of times in the next couple of weeks, and we'll talk more about what the research is. And most people get into science because they're interested in research, and that's the driving force, and then we do the other stuff sort of because you have to as part of the game. I'll give you a really quick summary of how I got here. So I went to high school. I was not a big fan of high school. My grades were pretty good, but I was not. It was pretty geeky, and I got beat up a lot. So that didn't make for high school being a lot of fun. So I went to university. I took 5 years to finish my university degree, which is okay. I took a year in the middle and traveled around a lot. I did a master's degree after that, and then I did a PhD, and then I did what's called a postdoc, and I worked in somebody else's lab. So I already had my PhD. I was doing research, but I was doing research in somebody else's group, and then I got my faculty job here in 2006. Okay, how old are you when you graduate high school?
18.
Yeah, so I was 18 when I graduated high school. 18 years later, I had my first job as a professor. So it is not a quick career path. It's an awesome career path. I honestly tell people that I have the best job in the world. But if you were looking for a job to sort of be gainfully employed soon after high school, being a professor is not it. I made $10,000 a year as a grad student for a long time, so it's not a quick path. Okay, so I mean that's sort of an overview, and if you have specific questions about what does going to university mean, if you're interested in a career of science, how do you do that? How do you decide you want to be a professor? There are lots of different answers to those questions that we can talk about. Let's talk a little bit about what I do as a researcher. So I'm a geneticist. I believe your teacher asked you to watch the TEDx talk that I gave last year. Is that right? Good show of heads, nodding. And how many of you actually watched the TEDx talk?
How many of you watched it?
A few hands. It truly is a wonderful and inspirational talk. So if you haven't watched it, I would suggest you do that. Okay. I'm a geneticist. And I think actually I start all of my talks saying that. I'm a geneticist. So somebody in the room, I'm going to put you on the spot. You in the yellow hoodie, orange topped hoodie, everybody's looking at you, so you're going to have to raise your hand. Yeah, what's genetics? So the question is, what is genetics?
DNA. There's substances contain DNA.
Yeah, so that's an awesome question, and I'm a fruit fly geneticist, and there are fruit flies that constantly fly around my office, and every now and then I forget the video camera's on, and so when I'm doing this, it's because I'm actually swatting the fruit flies. So that's an awesome answer, and it betrays the fact that you live in 2014, so you know what DNA is. How many of you, I assume now you have all heard of DNA. How many of you think you heard of DNA in grade school? Yeah, I talk to kids that are in 5th and 6th grade, and we talk about DNA. So DNA is really fairly a detailed part of genetics. Can somebody else give me a broader answer? What is genetics? And I'll give you a hint. Gregor Mendel was a geneticist, and he was doing genetics on peas, garden peas, 100 and something years before we knew what DNA was. So what do geneticists actually really study? Supergirl, come on. You're wearing the shirt. Step forward.
Is it the study of kind of what composes everything? Or I guess that would be cells, but what or how we're all different and what makes us different in our gene?
Yeah, so that's an awesome answer, and those two answers go together to give us a lot of genetics, but you're both missing one key feature. So the molecule of genetics is basically DNA, and that's not always true, but it's often true. What we're interested in genetics is what makes us, and all the complexity in us and our cells. Those are really great components. There's one crucial thing that makes genetics and not cell biology. What is it? Somebody else. The resolution is not going to allow me to pick on somebody other than just say, oh, yes, it will. You in the green striped shirt. What's one other piece?
Feel free to help her out, folks.
Like the chromosomes in the DNA, something? The
No,
differences between...
You're trying. It's awesome that you're all trying. The piece that you're missing here is the really obvious one, and you're just missing the trees to the forest. Genetics is about how this information is passed on generation to generation to generation. So it is all about DNA, and DNA makes up chromosomes, and that's the information that gives us that complexity that Supergirl was telling us about. But what that other component of genetics, and the thing that we're all really interested in geneticists, as geneticists, is how does that go generation to generation to generation? And that's what we study in my lab. So when I was first studying genetics as a master's student, I didn't do anything with DNA. We knew that the traits that we were studying were coded for by DNA, but we never looked at gene sequence. We were not following the chromosomes. We were just looking at the physical structures of these flies and how they were inherited generation to generation to generation. So that's really what defines genetics, and then you've got these other little pieces that make it up, like DNA. Okay, so that's a very broad definition of genetics. In my lab, what we try to do is connect biological complexity and genetic diversity. So we work on a number of different systems. Some of my graduate students work on bacteria. Some of my graduate students work on fruit flies. I have a new graduate student who started with me on Monday, and he works on cormorants, the birds that you see floating around in Lake Ontario, and we're trying to figure out what their diet is. All of those are the only unifying feature is that they're all biological systems, and we're looking at the genetics behind those systems. Okay. I can go into other little details, but I think that maybe you were tasked with trying to come up with questions for me. So do you have questions that we can start bouncing back and forth?
Yes, Dr. Merritt, we do.
Perfect answer. Way to go. Well, let's hear one of those questions by somebody that I didn't pick on before. If maybe one of the teachers would tap somebody on that, or point them out, or single them out, or... Good.
Okay. Jane Morgan, go.
Can cancer and other sicknesses be passed through the genetics in cells?
No. Are there any other questions? Okay, so that's an awesome question. All right, so the question was, give me the question again, and let's play with that. Give me the question again.
Okay. Can cancer and other sicknesses be passed through genetic cells?
Okay.
Or
So
genetics in cells, yeah.
Yeah, and let me see if I can paraphrase and say, do cancer or other diseases have a genetic basis? Is that a fair rewording of your question?
Yeah.
You don't think it is? That's okay. You can tell me though. So there are a lot of different answers to that. Okay, and I'll play with one of them, and then we'll bounce it back and forth. Can somebody tell me what cancer is? Don't say bad. I know it's bad, but what's cancer? Yeah, front.
Abnormal growth in cells.
That is an awesome answer. All right, so cancer is a sort of very broad term that we use for a series of diseases that are characterized by the abnormal growth of cells. That abnormal growth is either always genetics, or if it's not always genetics, I don't know that. So something goes wrong, and the cells grow at the wrong time, at the wrong place, with the wrong regulation. And that's the genetic basis of cancer. Now, I'm sort of avoiding your question in that there are a lot of different pieces. So is there a genetic component to disease? Almost certainly yes, but it depends on the disease you're talking about. Is there a genetic component to cancer? Cancer is a class of diseases. Sort of by definition, there's a genetic component to all cancers. But if cancer is the abnormal growth of cells, and that abnormal growth of cells has a genetic basis, do you think it's always going to be the same genetics? If we're talking about ovarian cancer, breast cancer, colon cancer, brain cancer, leukemia, there are hundreds, if not thousands, of cancers. They all have a genetic basis, but the genetics behind each of those can be slightly different. Does that make sense? And so often what we're trying to do is figure out what component of the genetics has gone wrong in each of those diseases. All right, let's take one step back, and then we'll come back to the idea of cancer. So what's a gene? Can somebody define for me a gene? Don't say the girl in my other class, because I know her, and she's not the right answer. Thank you, that's a joke. Okay. Somebody else give me either a better joke or the definition of a gene. Silence.
Sherry.
Okay. I think genes are the DNAs that pass on the characteristics.
You were almost correct. And again, you're stuck in the fact that you live in 2014. Genes are not always DNA. Most genes are DNA. But the other part of your definition, so the two little ends of the definition were spot on. Genes are packets of information that transmit that information generation to generation to generation to generation. In general, genes are DNA, but they're not always. They're RNA viruses, and the genes in those cases are RNA. There's some interesting cases where proteins fold, and that protein folding is passed on generation to generation, and we could argue about what that counts as. But a gene is simply a packet of information, often DNA, that's passed on generation to generation. Okay, so that's a gene. How many genes are in your genome? So the genome is the sum of all the genes in your body. How many genes are in your body?
I think he told you during
Don't
the
say a lot. Don't say a lot. Say again?
25,000?
Yes. I'm sorry, did you say 25 or 45?
25.
25. Okay. So there are between 20 and 25,000 genes in the human genome. The sort of cool thing is we thought that there were 100,000 genes in the human genome, and we had that wrong, and that was one of the things I talked about in that TED Talk. Okay, so there are 25,000 genes in the human genome. There are hundreds of different types of cells and tissues in your body. I mean, if you do this, you're feeling skin cells, but you're feeling them with nerve cells. You're moving those fingers with muscle cells. You're driving that with energy that's coming from your liver, and you're driving that with oxygen that's being pumped through your circulatory system by a heart, by blood. Those are all specialized tissues and cells. What defines a skin cell as a skin cell and a muscle cell as a muscle cell is what genes are turned on or off in that cell. So there are 25,000 genes in every cell in your body, but there are only about 2,000 genes that are turned on, okay? So a skin cell is a skin cell because the right set of 2,000 genes are turned on in that cell. Somebody make a guess. What defines that abnormal growth in cancer? So the identity of a cell is defined by the genes that are turned on in that cell. What's the relationship between that simple fact and cancer being the abnormal growth of cells? Yeah.
Would the gene be compromised, or would there be more or less than?
Okay, sorry, you cannot mumble because it then comes across as... So all I got was, would the gene be, and after that it was sort of downhill. So I think you're on the right track. Give me the answer again, but enunciate.
Okay. Would the gene have been compromised, or would there be more or less than there was supposed to be in a cell?
Yes. So, and this is the really... Okay, it's difficult to say the cool thing about cancer, but this is the cool thing about cancer. The unifying feature of cancers is that you've got a set of genes and the wrong gene is turned on at the wrong time. So what defines cellular identity, what defines tissues, is not the presence or absence of genes, it's which gene is turned on. All the genes are always present. It's just that some of them are on and some of them are off. In a cancer cell, the wrong set of genes are turned on or turned off at the wrong time. So is there a genetic basis to cancer? Yeah, by definition, that's what's driving cancer. The exciting thing from a geneticist's point of view is it means that as we begin to understand genetics, we have a better chance of understanding what's driving cancer. So if you want to cure cancer, it's not about treating the symptoms. If you want to make people that have cancer right now feel better, you treat the symptoms. If you want to cure the disease, you understand what's driving that disease. Why are those genes turned on at the wrong time, at the wrong place? Okay? What we do in my lab is try to figure out how those genes are turned on or off just in general. Okay? Now, why do I do it in fruit flies?
Oh, I'll do this one.
Okay.
Because they have a really short lifespan.
That is actually one of the really good reasons. So there's sort of, I can't remember, I should know this off the top of my head, but there are like 3 or 4 really good reasons. So we do this in fruit flies because they have a short lifespan, and that means that in the space of 2 weeks, I can have the flies that I'm interested in having. Why else? How big are fruit flies?
Small.
Small. So I can put thousands of fruit flies on my desk. And when you're working on genetics, the key feature in genetics is often genetic diversity. How different is one line from another from another? If I'm working on zebras, I can only get about 1.5 zebras in my office. There's not a lot of genetic diversity in 1.5 zebras. But if I can, in the space of my office, put 10,000 flies, that's a lot of genetic diversity. Why else? What would you do in fruit flies that you would probably not do to the person sitting next to you?
Kill them?
There is the kill 'em aspect. Yeah, I wasn't going with kill 'em, but we'll go with kill 'em. So I can sacrifice thousands of flies, and I'm not losing sleep over that. That is a good point. And whoever was sitting next to that person, you should feel reassured that they're unlikely to kill you this afternoon. So we work with flies. I mean, they are invertebrates. They're a really simple system. There are not a lot of ethical issues with working with vertebrates. They're small. We can keep thousands of them. We get a new generation of flies every 2 weeks. And all of those make it a really simple system to work with. But what is it about flies that allows me to understand humans? Go ahead with you. If you've got an answer in the front, don't worry about my follow-up question. What were you going to say?
Do they just do they age quickly?
They do actually age quickly, and I have avoided working on aging because there are 10,000 people working on aging. But we actually do some studies specifically on aging in flies because there's such a good system to work on. All right. The other piece of this, I want to understand cancer in humans. I don't actually, but let's say pretend. I want to understand the genetics of everything, humans being one of them. Why am I studying fruit flies? Because the genetics of fruit flies tells me a great deal about the genetics of human beings. We're all evolutionarily related. The way that genetics, the way that biochemistry, the way that DNA, the way that proteins, the way that chromosomes work in flies is similar or identical to the way it works in humans. And so by studying flies, I can better understand humans. In many cases, there's a huge overlap in the genes. So humans have about 20 or 25,000 genes. Flies have about 15,000. In many diseases, as much as 80% of the genes that we know are involved in the disease in humans are found in fruit flies. So you can study fruit flies because they tell us so much about how things work in humans. They're short-lived, they age quickly, you can keep lots of them. The other thing is we do a lot of genetic engineering, and this is actually where I was going, not that you're not going to kill the person next to you, but you're unlikely at the point now to genetically engineer your little brother. Now, at some point, you might be able to genetically engineer your little brother, but we can genetically engineer the flies, and we can go in and test things. If all you can do is observational biology, you're looking at correlations between diversity here and biological complexity. With things like fruit flies, we can genetically engineer it. So if we see a correlation between when this gene is turned on or off and abnormal growth of cells, that's a correlation. But we can predict that that's driving the cancer that we're seeing. We can go in and genetically engineer that gene in flies and test that prediction. And that's one of the major reasons that we work in any model system, whether it's mouse or rats or pigeons or flies. I just happen to work on flies because they're super easy and there are no issues with working with vertebrates. Okay, so that was a rather long-winded question or answer to your question, but that's what we're getting at. So is there a genetic basis to disease? Yeah. Is it complicated? It's incredibly complicated, and it's going to vary from disease to disease to disease. Okay, I think we've got about time for one more question if we want to bounce one more question, and then we will talk about our next meeting.
How about Abby? Abby, what do you guys have?
How do you modify genes?
The way that I do it is I go on my computer, and I call up the Drosophila Stock Center in Bloomington, Indiana, and I say, I'm interested in malic enzyme. What do we have in the stock center? And I order those flies that somebody else has genetically engineered. So one of the other reasons that we work on flies is that thousands of people have been studying flies since the early 1900s, and we have incredible resources that are out there so that you can take advantage of the fact that a lot of people have been working on this system before. The way that it's done, the way that genetic engineering is done initially varies on the organism that you're interested in, the type of mutation that you want to do, and the kind of questions that you're trying to ask. So the genetic engineering that we do in my lab is pretty straightforward, and will be interesting to explain in 4 minutes. So let's see what we can do. Basically, what we do is using a set of crosses where we cross this fly to this fly, we cause a piece of the genome to pop out. And when that piece pops out, it causes a mutation. And so this is called an excision-based mutation. It is not very exact, but it's super easy, and it takes zero equipment. And we can create a fairly broad suite of mutations that way. What we would really like to be able to do is literally program exactly what the mutation is that we would like to have happen, set things up so that that mutation is inserted into the genome, and then carried generation to generation to generation. And that is a surprisingly difficult thing to do. There were a set of tools that have been announced, published, made available in the last year or so that are getting us closer to that. And actually, you know what? I will make a note for myself. There were a couple of nice articles about this stuff. It's called the CRISPR-Cas9 system, and the name isn't important, but the concept is that basically we're getting closer to literally being able to program exact changes into the genome. And there were some interesting articles in the popular science literature, like the New York Times, on this genetic engineering system in the last year, because I talked about it with the undergrads that I teach in one of my classes. So I will see if I can dig up those references and send it to your teacher. So let's leave it at that. There are lots of different ways that you can do this. A lot of what we do is we cause random changes, and then we try to select out of those thousands of flies that we've randomly changed, which are the few that actually have the mutation that we want. What we'd like to be able to do is literally program in a change, and we're getting close to that. We're not entirely there. Okay, I've got 2 minutes and I have to run. We're going to meet again next week. What I would like you to do is actually, Tim, help me out. Where did we leave things for our meeting next week? What was the task that we were going to leave with, or we were going to challenge the students with?
Well, we were going to throw it back onto the class that next time we meet, some subset of the group, or perhaps the whole class, would in fact begin the discussion, much like you did today, telling us about you and your work, that they would find an area that they are interested in and learning more about, and perhaps put you on a spot a little bit more, but they would take the lead and perhaps tell you a little bit more about what they've understood and begun to learn as we delve deeper into our bio unit. By this time next week, they will be biological experts. They will have a very sound grasp of mitosis. They will work further into just what exactly is cancer, and they'll be becoming experts at least on 3 of the human systems. So I think between that background content and the curiosity that you've certainly spurred in them today, that we'll come forward with some topic that we'd like you to delve further in with us, but we will take the lead.
Okay, that's awesome. So if we can do that, and I don't know if it would be possible to work in like pairs or trios, there's no way I'm going to get through 12 questions, but we might be able to get through 4 questions or 6 questions. And I think we're going to have a third session after that. So let's see how far we get next week, and then we'll go from there.
Yeah, I was envisioning that they would kind of self-select a topic and develop their area of expertise, and therefore drive the discussion per their topic. So probably no more than 4 groups. Probably be more like 3.
Okay, that's awesome. That's fantastic.
Perfect.
Okay, folks, thanks for taking the time to chat with me this morning. We have our task. You're welcome. We've got our task for next week. We can go off task. So if there's some things that, you've got an opportunity to chat with a professor, If there's some questions that you want to ask that are outside of what we've talked about now, I mean, general things about trying to go to university or university classes, a career path, that kind of thing, bring them to your teacher and just give us a heads up, and we'll try to make some time to chat about that too.
Cool.
Thank
All right.
you
All
so
right.
much.
Dr. Merritt.
Have a great day.
Bye.
Have a great day.